आज के इस विश्लेषण में, we're taking a high-energy deep dive into one of the absolute most incredible yet totally unsung heroes of your entire body, the biological waste elimination system. I mean, think of your body as this massive, endlessly bustling factory. And look, just like any factory, creating energy and building products is going to generate trash. It's inevitable, and if that trash doesn't get taken out, the factory shuts down permanently. So today, we're going on a tour of the ultimate biological waste management facility to see exactly how your body stays alive. But you might be wondering, just how dangerous is this biological trash really? Well, take a look at this mind-blowing stat. Ammonia, which is just a totally common byproduct of our cells, is 100,000 times more toxic than urea. 100,000. It's kind of like, you know how you have to cut toxic people out of your life to protect your mental health? Well, our cells have to aggressively cut out these toxic byproducts just to physically survive. If this stuff accumulates, it is literally game over. Okay, let's dive right into our 6-part roadmap for this explainer. First, the toxic trash problem. 2, animal kingdom waste management. 3, human filtration machine. 4, the microscopic heroes. 5, the juxtaglomerular apparatus. And finally, 6, mechanics of ultrafiltration. Starting off with Section 1, the toxic trash problem, specifically metabolic byproducts. Because literally every single second of every single day, your cells are super busy breaking down nutrients to create energy. These metabolic activities are absolutely essential, but they inevitably create some really dangerous byproducts that absolutely must be expelled. So, here are the major culprits our cells are producing. We've got nitrogenous wastes, things like ammonia, urea, and uric acid, alongside carbon dioxide, excess water, and various ions like sodium and potassium. Whether it's just from normal daily metabolic activity or maybe, eating a bit too much of certain things, these substances just keep piling up. And if they just sit there, they completely hinder the cell's ability to function. So they've got to be removed either partially or completely. Which brings us to Section 2, Animal Kingdom Waste Management and the evolution of excretion. Because nature handles these exact same toxins quite a bit differently depending on where an animal lives. And the absolute biggest deciding factor here, how much water they actually have available to flush their system. Now, what's really interesting about this is how brilliantly adaptation actually works in practice. So if an animal lives in the water, like say a bony fish or an aquatic amphibian, water is obviously super abundant, so they can easily afford to use maximum water to flush out that highly toxic ammonia we talked about. We call them ammonotelic. But if you're a bird or a reptile, you really need to conserve water, right? So you convert your waste into uric acid, which is the least toxic option and requires the absolute minimum amount of water to get rid of. And then there's us humans. We, along with other mammals, sit right in the middle as ureotelic animals. We excrete moderately toxic urea using just a moderate amount of water. And as we trace this evolutionary timeline, we can actually see these early biological filters developing. Really simple flatworms, for example, they use these cool structures called flame cells. Earthworms have nephridia. Prawns use green glands. And insects, like your average cockroach, they rely on Malpighian tubules. But the big takeaway here is that all of these structures, no matter how different they look, perform the exact same crucial task of osmoregulation, which is balancing water and ions while simultaneously eliminating waste. All right, moving on to Section 3, human filtration machine, the kidneys. That whole evolutionary journey we just looked at, it brings us right to our highly advanced vertebrate setup. I'm talking, of course, about the human kidneys. Let's look at the physical specs really quick. You've got this pair of reddish-brown, bean-shaped organs sitting closer to your back, safely tucked right between your last thoracic vertebra, T12, and your third lumbar vertebra, L3. Now, they aren't massive. Each one weighs just about 120 to 170 grams and is roughly 10 to 12 centimeters long. But seriously, do not let their small size fool you for a second. Because if we were to slice a kidney wide open, we'd see this incredibly distinct macro architecture. On the very outside, safely protected by a tough renal capsule, is the outer cortex. Moving inward, we find the inner medulla. But here's the thing, the medulla isn't just some empty space. It's actually divided into these highly organized, distinct, conical masses that we call medullary pyramids. And these outer and inner layers actually interlock with one another. The outer cortex doesn't just sit on the surface. It actually extends downwards, weaving right between those conical medullary pyramids. These extensions are structurally vital to the whole setup, and they have a very specific name, the renal columns of Bertini. Okay, Section 4, the microscopic heroes, the nephrons. So now that we have the macro view down, let's zoom our perspective all the way in. It's time to meet the microscopic units that are actually doing all the dirty work. Just look at this number for a second. 1 million. That is the staggering number of incredibly complex tubular structures packed into just one single kidney. 1 million. These structures are called nephrons, and they are the true, undisputed functional units of your entire excretory system. Every single drop of filtrate takes a highly specific journey through these nephrons. It starts its trip in a little cup-like structure called Bowman's capsule, moves into the highly folded proximal convoluted tubule, and then dives way down into the loop of Henle. And the loop of Henle is super recognizable because it has a very distinct hairpin shape. Honestly, it looks exactly like a U-pin or, a Judah pin. After coming back up from that dive, the fluid travels through the distal convoluted tubule before finally draining out into a shared collecting duct. And this brilliantly illustrates exactly how our bodies optimize resources. Get this. 85% of our nephrons are what we call cortical nephrons. They have really short loops of Henle and stay mostly up in the outer cortex. But those remaining 15%? Those are the juxtamedullary nephrons. These guys have massive, deep-diving loops of Henle that plunge straight down deep into the medulla. These heavy-duty nephrons are kept in reserve for when your body really needs to seriously conserve water and concentrate your urine. All right, Section 5, the juxtaglomerular apparatus, or JGA, the regulatory sensor. Because, as you can imagine, to manage all of this insanely complex microscopic plumbing, the nephron needs a highly sensitive, built-in control system. And here's how that works. Because of how the nephron literally folds in on itself, the distal convoluted tubule actually loops back around and physically touches its own incoming blood vessel, the afferent arteriole. And when these two specific structures touch, something kind of amazing happens. They undergo cellular modifications to create this crucial regulatory sensor. The blood vessel's smooth muscle form what are called JG cells, which can secrete a vital hormone called renin. Meanwhile, the tubule cells pack super tightly together to form the macula densa. Working together, they basically act as a highly sensitive, real-time flow meter for your kidney. Which brings us to our grand finale, Section 6, the mechanics of ultrafiltration, the squeeze. We've got the structure, we've got the control center. Now let's look at exactly how the blood is physically squeezed to actually remove all that trash. As blood rushes into the nephron, it is forcefully pushed through a microscopic 3-layer barrier. First up is the inner lining of the blood vessels. Second is a basement membrane right in the middle. And third, you have these highly specialized cells on Bowman's capsule called podocytes. These podocytes are really cool because they have tiny little gaps between them known as filtration slits. This entire 3-layer barrier basically acts like a perfect microscopic sieve. It allows the liquid plasma and all the small waste molecules to push on through, but it safely leaves the larger proteins and blood cells behind in the bloodstream where they belong. So the crucial point here is understanding the physics of why the fluid actually pushes through. It's essentially a massive battle of pressures. The sheer pressure of the blood rushing in pushes outward at a positive 60 millimeters of mercury. But it faces resistance. The proteins that were left behind in the blood, they're pulling back with a negative pressure of 30, and the fluid that's already sitting in the capsule is pushing back with a negative pressure of 20. But when you do the math, the incoming positive pressure still wins by a net of plus 10. And that net positive pressure of 10, that is the relentless driving force that filters your blood every single second of your life. And the sheer scale of this operation is, frankly, almost unbelievable. Because of that constant, relentless pressure we just talked about, your kidneys generate 180 liters of raw filtrate every single day. 180 liters. That breaks down to about 125 milliliters every single minute. But that leaves us with a massive mystery to wrap up this explainer. If our microscopic heroes are producing 180 liters of raw filtrate every single day, why do we only actually excrete about 1 to 1.5 liters of urine a day? I mean, where exactly do the other 178.5 liters go? Just think about the sheer biological marvel of a system that selectively reabsorbs 99% of what it filters, saving all the good stuff and tossing only the actual trash. It is a completely incredible story of survival and efficiency, and for sure, it's a topic we're going to tackle in our next deep dive. Thanks so much for exploring the biological factory with us today.